About a year ago, I looked in my tank and saw what all shrimp keepers dread. Tiny, wriggling, hair-like parasites on top of one of my shrimp's heads. After a quick Google search, I discovered that these hairs were the start of a slow but dangerous infestation of an organism known as Scutariella japonica, a worm that reproduces in the gills of shrimp and chokes off water flow, eventually killing their host. I was scared and wanted to learn how to save my shrimp, how to protect them. But I was also curious about what kind of a creature S. japonica is, how its life cycle works, how it got in my tank. So I did some research. After hours sorting through scientific papers, and thanks to the hard work of scientists like Rafal Masiacek, my perception of S. japonica completely flipped, to the point where I don't even think they're parasites in most circumstances. In fact, they may even be beneficial to our shrimp. Isn't that crazy? The research also gave me a new appreciation for how much we still don't know and are learning as the science continues to progress and as this hobby continues to progress. Hopefully, by the end of this video, you get a little bit of that appreciation too. So, if you're in the position that I was a year ago, and you see those little white worms on your shrimp, you look it up online, you start getting worried, just know you can relax a little bit. In many cases, S. japonica and the organisms that are commonly mistaken for it are actually perfectly harmless and may even be beneficial. That said, certain conditions do make them parasitic and dangerous, and that's where we should be concerned. The rest of this video is dedicated to understanding these organisms more, how they interact with our shrimp, and what conditions they become dangerous. Let's get started by understanding exactly what a parasite is. Parasitism is the association between two different organisms, wherein one benefits at the expense of the other. Keep in mind that this refers to the overall impact of an organism, meaning even if an organism does cause some harm, it is not considered parasitic if the benefits outweigh that harm. One example might be a tapeworm in your gut. It benefits by absorbing the nutrients that you've eaten while you lose those nutrients. Another example might be the hated mosquito, which sucks up the blood that you worked really hard to make and potentially gives you something like malaria in return. Mosquito benefits and you do not at all. It sucks. Definitely a win-lose situation there. While we traditionally think of these relationships as fairly fixed things, that something that is parasitic will always be parasitic, that's not always the case. One example of this is the symbiosis between coral and the algae inside of it. Under normal conditions, the coral provides protection and resources for the algae, while the algae in turn provides oxygen and other nutrients that the coral needs to grow. So they both benefit in this relationship. Things change though when the oceans warm, as we're seeing with human-caused climate change. In even slightly warmer water, the algae starts to produce byproducts like hydrogen peroxide, which damage the coral cells. The algae continues to take coral resources and produce damaging compounds until the coral eventually rejects it in a process known as coral bleaching. So that's just one example of how the relationship between organisms can change depending on the environment, but I think you get the idea. Now let's go more into shrimp and organisms like S. japonica. Instead of calling them parasites, we're going to refer to them as epibionts, or an organism that lives on another organism. So what are these little white hairs on our shrimp? As I mentioned in the introduction, these tiny white hairs you see on your shrimp may actually be one of three organisms. The first is S. japonica. It falls into a family of flatworms known as Temnocephalida. S. japonica has a sucker on one end to attach to the surface of the shrimp and has two tentacles on the other for catching small microorganisms. The next is Vorticella, which is technically a group or a genus of multiple species. These are single-celled protozoans with a feeding end shaped like a bell and a stalk that attaches to different surfaces. They eat other small organisms, if you've played Pokemon, kind of like a very small weeping bell. The third is Monodiscus kumaki. This is a type of rhabdocella worm with a sucker on one end that attaches to surfaces and two stubby tentacles on the other. It uses these tentacles, just like S. japonica, to eat microorganisms like rotifers and protozoans. Now, how does the presence of these epibionts affect your shrimp? For one, the suckers of S. japonica and M. kumaki do cause shallow mechanical damage wherever they latch on. This may appear as tiny cracks in the shell or discolored spots on the gills. This mechanical damage has not been shown to affect mortality and disappears after a molt. The second way that S. japonica and M. kumaki affect your shrimp is that they lay their eggs inside the gills. This means that the eggs and any large population of adults may clog the gills and cause problems for your shrimp. The third way their presence affects your shrimp is that these epibionts eat microorganisms found in the gills and on the surface of your shrimp. While we don't have studies specifically about the impact of any of these particular species on Neocaridina, we do have studies on closely related species that can help us hypothesize their impact. In 3 different studies, researchers found that these closely related worms had a strong effect on the growth rate of their host crayfish. They found a parabolic relationship between the quantity of worms present and the crayfish growth rate, meaning the crayfish grew better when some worms are present compared to either none or high densities. The researchers hypothesize that at low to moderate densities, the worms have a mutualistic relationship where they clean debris or potentially harmful epibionts from the gills. So these worms have a similar diet to S. japonica, where they feed on other bacteria and microorganisms that may end up in the gills of these crayfish and could potentially cause infection or clog the gills. So they are effectively helping to clean the gills. At higher densities, it appears that they shift to a parasitic relationship because there may not be enough food to share in the gills, leading the worms to feed on the gill tissue and reducing the growth rate of these crayfish. Again, while there are no studies specifically on S. japonica, Vorticella, or M. kumaki, it's not unreasonable to assume that these worms behave in a similar manner. I actually emailed the head researcher and got a chance to talk to him briefly based on what he's seen with the impact through hundreds of shrimp and his own shrimp breeding experience. He no longer treats his shrimp for S. japonica because there's not really any evidence that they cause harm at low levels. And while this is some anecdotal evidence, my experience supports this. The small white hairs that I see on my shrimp are M. kumaki, as confirmed by the leading researcher. While I do see an occasional hair or two on my shrimp, they never reach high densities that could become problematic, and so I leave them. So when do these organisms become parasitic? These epibionts only overpopulate when they have enough food. Their food is debris and microorganisms in the water. If you have a lot of that in your water, then your water quality is not good. Your shrimp are also suffering because all of that stuff is ending up inside of their gills. These epibionts may in fact be benefiting your shrimp in this environment, reducing the impact of bad water quality. This actually makes these species good indicator species for the overall health of your aquarium. Because even if your nitrates, your ammonia, your pH, everything seems to be in balance, you can still have bad water quality due to all of the microorganisms and debris in the water that don't show up on those tests. Again, I know that M. kumaki is present in my tank, but I almost never see it. The only time I do start to see them is after disturbing the substrate, after I've thrown up a bunch of debris in the water and these organisms now have a food source available, because the water quality isn't as good as it was. As soon as I give this debris time to be filtered out and to settle back, those hairs disappear. So my experience supports the hypothesis in this study, and now I actually like having them. I like knowing whether my water quality is good. That's basically what these are telling you. If you've noticed the same thing as me, or if you've had a different experience, then I'd love to hear about it in the comments. Let's have a discussion. Let's learn more and move the shrimp keeping hobby forward. Because if we find that having the epibionts on our shrimp potentially increases growth like we see in the crayfish study, that could be really useful for breeding. It's just so cool to think about how much there still is to learn. This all begs the question of, when is treatment required? Now, let's go into that. If you do have shrimp with a lot of white hairs on them, that's a sign of a much larger problem with your tank that is impacting your shrimp. These organisms are telling you that something is wrong. And so you have 2 options. One, you can treat your individual shrimp, you can treat your tank, and yes, it will kill these organisms. It will also kill some of the microorganisms that may be in the water and could improve water quality a bit. It isn't addressing the larger problem of why your tank has bad water quality in the first place. So you are effectively treating a symptom of a larger problem that may lead to more issues down the line. Again, even if your parameters say it's good. That's because parameters only test for certain chemical properties of your water and do not provide like a holistic look at your ecosystem. Understanding water quality, what causes bad water quality, how to fix it, is really a whole nother topic, and we cover that in an article that was recently released on the website. I highly recommend going and looking at that article and seeing if any of the examples that we provide for what causes bad water quality are potentially problems with your tank. Once you solve that problem, then your epibiont problem, it's going to go away. They're still going to be present in your tank in very small quantities to tell you if anything like this happens again. So that's really, really useful. But again, as we've mentioned, large populations are problematic. So if you do see them on your shrimp, you may want to treat them individually or at a tank level. We don't have research to say exactly how many become harmful, but I'd say if you're seeing like 10 or more hairs, or you're seeing a good fuzzy patch around their head from Vorticella, then it's a good sign that you may want to treat that individual or your tank as a whole. The easiest way to treat individuals is to do a salt dip. The research recommends a 30-second dip in tank water with 40 grams of sodium chloride per liter. If you have multiple shrimp with a lot of Epibionts on them, then you may want to consider doing a whole tank-wide treatment using hydrogen peroxide. Mark's Shrimp Tank has a great video showing how to do this treatment up here, and it'll also be linked down below. You can also check out our article to see text-based instructions if you prefer that. The truth is, if you set up and care for your tank properly, then Epibionts aren't going to be an issue, and neither is bad water quality. Check out this video to learn more about what goes into a healthy shrimp tank and how to know when your tank is actually ready for shrimp, or you can check out simplyexplained.com for more information on a variety of topics. I'd also like to give a shout out to Rafal, who is the head researcher of this paper, for doing more research into shrimp diseases and for the kumaki project. This project helps to bring more attention to shrimp epibionts like S. japonica and their potential impact on both our shrimp and the environment. This is especially important because Neocaridina, unfortunately, are becoming invasive species in areas like Hawaii, which means they're potentially impacting the local populations in a negative way, and S. japonica or other organisms that they may carry could also have an impact. There's so many different interactions that we can't possibly predict. So please do not release your live or dead animals into the wild so we can reduce the impact of invasive species and protect our native habitats. Thank you in advance for protecting natural beauty around us, and I hope you have a great rest of your day. Keep learning.